8.3 Using light energy to make organic molecules. By the end of this section, you will be able to do the following: describe the Calvin cycle, define carbon fixation, explain how photosynthesis works in the energy cycle of all living organisms after the energy from the sun is converted into chemical energy and temporarily stored in ATP and NADPH molecules. The cell has the fuel needed to build carbohydrate molecules for long-term energy storage. The products of the light-dependent reactions, ATP and NADPH, have lifespans in the range of millions of seconds, whereas the products of the light-independent reactions, carbohydrates and other forms of reduced carbon, can survive almost indefinitely. The carbohydrate molecules made will have a backbone of carbon atoms. But where does the carbon come from? It comes from carbon dioxide, the gas that is a waste product of respiration in microbes, fungi, plants, and animals. The Calvin cycle in plants: carbon dioxide, CO2, enters the leaves through stomata, where it diffuses over short distances through intercellular spaces until it reaches the mesophyll cells. Once in the mesophyll cells, CO2 diffuses into the stroma of the chloroplast, the site of light-independent reactions of photosynthesis. These reactions actually have several names associated with them. Another term, the Calvin cycle, is named for the man who discovered it, and because these reactions function as a cycle. Others call it the Calvin-Benson cycle to include the name of another scientist involved in its discovery. The most outdated name is dark reaction, because light is not directly required, figure 8.17. However, the term dark reaction can be misleading because it implies incorrectly that the reaction only occurs at night or is independent of light, which is why most scientists and instructors no longer use it. 8.3. Using light energy to make organic molecules. 217 Figure 8.17 Light reactions harness energy from the sun to produce chemical bonds, ATP, and NADPH. These energy-carrying molecules are made in the stroma where carbon fixation takes place. The light-independent reactions of the Calvin cycle can be organized into three basic stages: fixation, reduction, and regeneration. Stage 1. Fixation in the stroma. In addition to CO2, two other components are present to initiate the light-independent reactions: an enzyme called ribulose 1,5-bisphosphate carboxylase, oxygenase, Rubisco, and three molecules of ribulose bisphosphate, RUBP, as shown in figure 8.18. RUBP has 5 atoms of carbon, flanked by 2 phosphates. Visual connection. Figure 8.18 The Calvin cycle has 3 stages. In stage 1, the enzyme Rubisco incorporates carbon dioxide into an organic molecule, 3-PGA. In stage 2, the organic molecule is reduced using electrons supplied by NADPH. In stage 3, RUBP, the molecule that starts the cycle, 218 Chapter 8, Photosynthesis access for free at openstax.org, is regenerated so that the cycle can continue. Only one carbon dioxide molecule is incorporated at a time, so the cycle must be completed 3 times to produce a single 3-carbon G3P molecule, and 6 times to produce a 6-carbon glucose molecule. Which of the following statements is true? A. In photosynthesis, oxygen, carbon dioxide, ATP, and NADPH are reactants. G3P and water are products. B. In photosynthesis, chlorophyll, water, and carbon dioxide are reactants. G3P and oxygen are products. C. In photosynthesis, water, carbon dioxide, ATP, and NADPH are reactants. RUBP and oxygen are products. D. In photosynthesis, water and carbon dioxide are reactants. G3P and oxygen are products. Rubisco catalyzes a reaction between CO2 and RUBP. For each CO2 molecule that reacts with one RUBP, 2 molecules of another compound 3-phosphoglyceric acid, 3-PGA, form. PGA has 3 carbons and 1 phosphate. Each turn of the cycle involves only one RUBP and 1 carbon dioxide and forms 2 molecules of 3-PGA. The number of carbon atoms remains the same. As the atoms move to form new bonds during the reactions, 3 C atoms from 3 CO2 plus 15 C atoms from 3 RUBP equals 18 C atoms in 6 molecules of 3-PGA. This process is called carbon fixation, because CO2 is fixed from an inorganic form into organic molecules. Stage 2. Reduction ATP and NADPH are used to convert the 6 molecules of 3-PGA into 6 molecules of a chemical called glyceraldehyde 3-phosphate, G3P. That is a reduction reaction because it involves the gain of electrons by 3-PGA. Recall that a reduction is the gain of an electron by an atom or molecule. 6 molecules of both ATP and NADPH are used. For ATP, energy is released with the loss of the terminal phosphate atom, converting it into ADP. For NADPH, both energy and a hydrogen atom are lost, converting it into NADP+. Both of these molecules return to the nearby light-dependent reactions to be reused and re-energized. Stage 3. Regeneration. Interestingly, at this point, only one of the G3P molecules leaves the Calvin cycle and is sent to the cytoplasm to contribute to the formation of other compounds needed by the plant. Because the G3P exported from the chloroplast has 3 carbon atoms, it takes 3 turns of the Calvin cycle to fix enough net carbon to export 1 G3P. But each turn makes 2 G3 petaseconds, thus 3 turns make 6 G3 petaseconds. 1 is exported while the remaining 5 G3P molecules remain in the cycle and are used to regenerate RUBP, which enables the system to prepare for more CO2 to be fixed. 3 more molecules of ATP are used in these regeneration reactions. Link to learning this link, http://openstax.org/l/Calvin_cycle leads to an animation of photosynthesis and the Calvin cycle. Evolution connection. Photosynthesis. During the evolution of photosynthesis, a major shift occurred from the bacterial type of photosynthesis that involves only one photosystem and is typically anoxygenic, does not generate oxygen, into modern oxygenic, does generate oxygen, photosynthesis, employing 2 photosystems. This modern oxygenic photosynthesis is used by many organisms, from giant tropical leaves in the rainforest to tiny cyanobacterial cells, and the process and components of this photosynthesis remain largely the same. Photosystems absorb light and use electron transport chains to convert energy into the chemical energy of ATP and NADH. The subsequent light-independent reactions then assemble carbohydrate molecules with this energy. In the harsh dry heat of the desert, plants must conserve every drop of water must be used to survive. Because stomata must open to allow for the uptake of CO2, water escapes from the leaf during active photosynthesis. Desert plants have evolved processes to conserve water and deal with harsh conditions. Mechanisms to capture and store CO2 allows plants to adapt to living with less water. Some plants such as cacti, figure 8.19, can prepare materials for photosynthesis during the night by a temporary carbon fixation, storage process, because opening the stomata at this time conserves water due to cooler temperatures. During the day, cacti use the captured CO2 for photosynthesis, and keep their stomata closed. 8.3. Using light energy to make organic molecules. 219 Figure 8.19. The harsh conditions of the desert have led plants like these cacti to evolve variations of the light-independent reactions of photosynthesis. These variations increase the efficiency of water usage, helping to conserve water and energy. Credit: Peter Wojtkowski. The energy cycle. Whether the organism is a bacterium, plant, or animal, all living things access energy by breaking down carbohydrate and other carbon-rich organic molecules. But if plants make carbohydrate molecules, why would they need to break them down, especially when it has been shown that the gas organisms release as a waste product, CO2, acts as a substrate for the formation of more food in photosynthesis? Remember, living things need energy to perform life functions. In addition, an organism can either make its own food or eat another organism. Either way, the food still needs to be broken down. Finally, in the process of breaking down food, called cellular respiration, heterotrophs release needed energy and produce waste in the form of CO2 gas. However, in nature, there is no such thing as waste. Every single atom of matter and energy is conserved, recycled over and over infinitely. Substances change form or move from one type of molecule to another, but their constituent atoms never disappear. Figure 8.20. In reality, CO2 is no more a form of waste than oxygen is wasteful to photosynthesis. Both are byproducts of reactions that move on to other reactions. Photosynthesis absorbs light energy to build carbohydrates in chloroplasts, and aerobic cellular respiration releases energy by using oxygen to metabolize carbohydrates in the cytoplasm and mitochondria. Both processes use electron transport chains to capture the energy necessary to drive other reactions. These two powerhouse processes, photosynthesis and cellular respiration, function in biological, cyclical harmony to allow organisms to access life-sustaining energy that originates millions of miles away in a burning star humans call the Sun. 220 Chapter 8, Photosynthesis access for free at openstax.org. Figure 8.20 Photosynthesis consumes carbon dioxide and produces oxygen. Aerobic respiration consumes oxygen and produces carbon dioxide. These two processes play an important role in the carbon cycle. Credit. Modification of work by Stuart Basile.